Hello, everyone, and thanks to the organizers for organizing this wonderful conference. It's a great pleasure to be here. And today I want to talk to you about how we can use or how we can actually leverage protein language models to figure out deep phylogenies and deep evolution of proteins. So the premise is that the whole thing that we're trying to figure out is how how organisms develop new traits and functions, which in our case, in case of molecular biology, is that how new genes come into being. So the theory behind is that they are mostly associated with duplications in terms of theory, and it all comes back to Suzumu Ono back in the 70s, where he said that genome duplication will spark novel adaptations. And essentially, this model boils down to just you have two genes, then you start with a single gene with one function, then you get a duplication, and in which case the selection on both of the genes is low because now you have two copies performing the same function, and one of those copies is allowed to mutate into a new function. Basically, every single other model is the variation of this model. So subfunctionalization is the same idea. The specialization is the same idea, but the question is like, do we even need bloody duplication? And like, there are ways to get new functions without new duplication. But the important point is that all of those models are purely theoretical. The amount of the things that we actually know about the evolution of new functions is extremely low. Not only we're dealing with the genes that are misannotated throughout. Every time I look into, the work that you guys are doing is great, but just bear in mind that UniProt is mostly garbage. And the genes that are annotated de novo without a human looking into them, every time I try to annotate a protein family, I would find that it's usually a ballpark of 40% of the genes being chimerically annotated, misannotated, missing, and so on. So when we take those genes, predict proteins from them, and then dump them in the UniProt, and then we train a fancy predictor on top, we're essentially feeding into our models the proteins that do not exist. And this is a very important point that we have to keep in mind. Like, we cannot overcome it right now because the only way to properly annotate genes is actually to go in there, look at the actual genomic code with your human eyes, figure out where are the exons, and figure out how you can make those exons into a proper gene model for a particular gene family. There is not a single tool that will allow you to do that in a batch size. You can predict only protein family by protein family. That's it. That's the cap. So that should not stop us, of course, from, doing large-scale analysis of the likes that we've just seen, which is, they are super valid, but they come with a lot of this, caveat that in practice, we have really no idea. So how do we do that if we really want to figure out it? So the good thing about my work is that I accidentally just, by virtue of my progression in science, I stumbled upon a very nice model, which is venom evolution. Because in majority of genes, the transition of a function is very hard to catch. While in the evolution of venom, evolution of toxins, they all evolve, protein toxins, of course, they all evolve from a, they call that in the field physiological gene, but we, of course, understand that venom is also physiological, right? So you start with a non-toxin gene, and then that non-toxin gene becomes a toxin gene. And usually you can easily catch it because that would change the species ecology, that would change the species morphology, and the structure of the protein is usually also quite visible. So in this situation, I have a very good model to try and figure out how the actual evolution of function happens, at least in that slice. So, but it doesn't really, stop there, because if you just take, for instance, if you have like a phospholipase here, right? If you look at the your databases and your genomes, you'll find that there will be a bunch of phospholipases for each given species. So you cannot establish orthology with the tools. You need to figure out the synteny, and only then you can figure out whether you're dealing with the direct orthologs or actually like quasi-orthologs. So in each case, you need to, figure out the synteny. So the way it works, for those of you who are not familiar, so if we take our genes and put them in the box and then look at our neighboring genes, then we trace those neighboring genes to the other species and we get a picture. So if we trace it back enough, and if we are lucky, we have enough of genomes to do that, then we will essentially hit a point where the gene this gene family does not exist in that particular locus of a genome. So in which case you can be safe that this everything downstream will be your orthologs, and you can study that. So in this particular case, in case of those phospholipases, what we saw is that in the amphibians, you have a single gene. Then between amphibians and amniotes, the gene does double triplication. So it triplicates once, then the triplication duplicates, and then you have a cluster with which all amniotes work. But the curious thing is that what we saw is that the theory predicts essentially that if you have a cluster of genes that are nigh identical, like the sequence identity there would be, sequence similarity there would be like close to 80%. So theory would say that every single gene of those is likely to duplicate and likely to create new functions as much as any other gene, unless you have some sort of a histones, changing that. But what we saw, and what I saw pretty much in every single protein family that I looked at, is that that's not the case. That all new functions in a given protein family usually evolves from a single gene or like neighboring genes, despite the neighbors being of the same structure. So in this case, we have this very stable situation where, except for amphibians, every single one has those 6 to 8 genes, but the new functions evolve only from the middle of the cluster, only from a single gene, independently in birds, from mammals, from snakes. And so, of course, my point of interest here were snakes, which are here. And so another cool thing is that usually the change of protein structure happens before the duplication, at least in this situation and some other situations that I saw. So it seems that there is, it basically hints, and I mean, I saw some of your posters that have seen somewhat the same thing, that we are yet to discover genomic regulations that are evading us so far, because it seems that, I mean, it kind of makes sense if you think about it, because the evolvability evolves. So the more you evolve, and the more complex you become, the better you will be at evolving. So it, makes sense that there are some genomic regulations that would create more likely points for the mutations and less likely points for mutations. And there is some sort of don't get me wrong, I'm not saying that there is a teleology to evolution, but there is definitely a understanding within the genomic structure, which genes we can tinker with, at least somewhat, and which genes we cannot tinker with. Another very important point here before we go into all the ML stuff is that if you are looking for complete proteins in your genome, you will not see stuff, because you need to be looking in the genome in the way the genes exist in the genome in eukaryotes. I'm talking about eukaryotes in this case. And in eukaryotes, they exist in exons. So if you're looking, if you're using anything but the exonic sequences themselves, you will naturally miss genes. For instance, in this case, they changed their annotations after we published the preprint, so they were a bit cheeky with that. But before this publication, if you looked at the crocodile, for instance, you would see that this gene is paired with this gene into a single protein. So instead of seeing 2 different genes, you would have one half from this gene connected to one half of this gene. We totally miss those 2 exons because we're not looking for exons. And so instead of having a proper structural things, you would have several genes concatenated essentially into chimera. And you can find that chimera in UniProt, and you can train your models on that chimera. So that's not really nice stuff. But it kind of gets even more complicated than this, because genes do not care for our bifurcating trees. Bifurcating trees is a hypothesis that we use to understand evolution. But if you have a triplication, that already kind of messes up with the bifurcating tree. But it gets even further. Like, for instance, let's see at the way we reconstructed the snake viperid phospholipase evolution. So if we start with the Colubroidae, which is your elapids, so your cobras, your vipers, and your colubrids, you would see that at the basis, there would be 4 genes. What happens next is very bizarre. We've only been able to trace it because I manually annotated 100 vertebrate genomes, right? Manually, every single gene. So what the reconstruction looks like is that you have a bunch of really bizarre duplications and really bizarre gene reduction and gene change. The interesting situation here is that independently, two lineages of snakes evolved dimeric neurotoxins. So they took this phospholipase that usually functions as a single, as a monomer, and they evolved to have additional cysteines that create a paired cysteines allowing for the dimer to happen. If you just use sequence homology, then you will naturally put everything that has those additional cysteines into a single group. However, our reconstruction shows that they evolved independently from different sublineages of genes. So if we just plot it as a tree or as a sequence similarity, we will get a wrong idea about the gene evolution in this case. We would miss a convergent evolution. So how can ML help us with this? Well, let's use a case study of three-finger toxins. Three-finger toxins is another group of snake venom toxins. The starting point here is that we know that there are three-finger toxins, and then they belong to a Ly6 group of receptor proteins, most of which are membrane-bound. So the difference between that and three-finger toxins and some of non-membrane-bound is that they are not membrane-bound. So in here you can see this is a three-finger toxin, and this is a membrane-bound Ly6. So it has an additional loop that is being clipped to allow for an attachment of the GPA anchor that will anchor the thing into the membrane and then stays there. So three-finger toxin somehow lost that. So what we did essentially, I mean, we collected all the genes in the aforementioned way, and then we also took all the experimentally validated proteins so that we can properly put a tree together. So that tree was a tree, right? But it still has a lot of kind of a small unresolved clades, and it has a lot of uncertainties that would be great to resolve. And it also doesn't really give us a good understanding of how those things happened in terms of the sequence of events. So we go back to synchrony and we trace all our genes. And if we just try to phylogenetically figure them out, we will be slightly confused because, I mean, they are weird. And so also another thing that is important is that if we just take model species and we start comparing model species to each other, then we will miss a lot of intermediate steps. So we need to have those intermediate steps to understand stuff. And if we, in this case, exclude snakes from the picture, then we would have pretty much the same situation as phospholipases. We would have a very stable cluster of protein families, right? So each gene in this situation, except for top one isomerase, so everything that is colored, is of the same protein family. They are all kind of like similar to each other. And they are extremely stable. We're talking about almost, 300 million years. So just by virtue of chance, they should be different, but they don't. So they don't really duplicate much. However, if we go into snakes, we will see a lot of crazy shenanigans happening. From 6 to 8 genes, we jump up to 30, and some snakes I've recently annotated had like 80 genes. And another interesting thing is that if we keep tracking those exons, we would see a lot of birth and death happening. So just figuring out the phylogeny on those things alone can get you quite crazy. And especially if you go and try to figure out all the pseudogenes where they're coming from, that would be an additional thing. But a cool thing that we saw in there is that each individual three-finger toxin, instead of having a standard 3 exons for Ly6, they had 4 exons, one of which they never used. So in each of cases, one of the gene has an exon that is always silenced and never used, but it has a enough of evolutionary information for us to figure out what it was there for. So how does ML comes into play? Well, I'm not going to bother you with the details of how the ML representation is created. It's just what is important is that we are working with a multidimensional vectors that are usually at the ballpark of 1024 or 2048 dimensions. Obviously, unless we want to work with them with multidimensional space, we need to somehow simplify it. So usually what we do is we use UMAP projections that would then collapse that thing to a 3-dimensional space and or 2-dimensional space, obviously with the loss of information, but trying to preserve as much of a structure there as possible. And once we visualize that space, we can see and work with it. But I mean, of course, if we can work with a multi-dimensional space just using Euclidean and cosine Euclidean distance to cosine similarities, then it is much better. So in case of our three-finger toxins, the UMAP projection looks like this. And what is really peculiar here is that it's essentially every functional cluster is separate. But another cool thing here is that we have all of the membrane-bound lysics making a kind of a tight cluster. Of course, if we remove all three-finger toxins and zoom on those, they would also cluster, more separately, more sparsely. But UMAP is taking into consideration the entirety of the data set, so that kind of changes stuff. With the three-finger toxins, though, we see how in this projection it kind of creates a separate space. So there is a jump from when we lose this membrane-bound anchor. So when we lose a membrane anchor and go into the secretory parts, from there we start exploring, and so the most toxic forms will be on the outskirts. So in here you have your cytotoxins, and here will be neurotoxins. And the interesting thing here is that there are a bunch of clusters that don't have a proper function annotated to them. However, when I talked to a few of the three-finger toxin researchers, which I have a luxury from being from that field previously, they told me that that makes much more sense. So, for instance, most of the three-finger toxins are NAV channel blockers, but this small cluster is a potassium channel blockers, of which, a few ones were tested, but the majority is not. However, they do make quite a tight cluster. So we already can see how this kind of a projection allows us to see stuff almost similar to sequence function exploration. Of course, everything here is an extent forms, right? So we obviously don't have the forms that do not exist. So for proper sequence function, we of course need them. So we only have the tip of the iceberg there. But it's still, it seems that we can map sequence function exploration with the extent forms already quite nicely. And so, the bottom line of this paper was that essentially this membrane anchor, that third silenced exon, was housing the original membrane anchor, and the duplication of that exon allowed for the evolution of new function. And what kickstarted the sequence function exploration by the three-finger toxins there. But also, the same stuff happened in humans totally independently. Yeah, this is just a pretty picture. But let me highlight you another thing that we were able to use it with. So there is a paper that claimed that a bunch of wasp, ant, and bee peptides all fall into the same clade. And the reasoning for that is essentially this figure. I promise you, they don't provide any statistical data. They just don't do anything else except for this alignment. This is the whole reasoning. And that alignment essentially postulates that despite the dissimilarity of the sequences, which if you build a tree of, you will see that they're highly dissimilar, the propeptide and signal peptide are similar enough to say that they all evolved from a single ancestor. So, we wanted to check it. So as a part of our paper on bee venom evolution, we decided to check that stuff. So again, I annotated at this point close to 100,000 genes. I'm not kidding, manually. Not all of them made into the paper, but I had entire 2 years of COVID to do that, so that's what I did. And in there, what we see is that if we take them now by protein families, not only they will cluster nicely together if we put all of them in one space, they would cluster by protein family. But if we go into each individual family, then subfamilies of that thing would still cluster together, right? So in this case, the coloring by the subfamily is derived from the phylogenetic tree that we built with Exabase. So you can see how the tree phylogeny maps, in some cases, not exactly, because obviously the protein language model doesn't have just the evolutionary information in it. It has gazillions of stuff, who knows what. It's still a black box for us. But it clearly has evolutionary information enough to separate the subfamilies of each of the protein family, which we see consistently. However, if we go into those aquila toxins, we see that they not only start invading into the space of the other protein families, they also seem to be clustering mostly by species and not by protein families. And so when we try and embed those proteins with signal peptide, we actually see the reverse of the claim. We actually see that they cluster by species much better than they cluster you can see a clear separation by species. And when we remove the signal peptide, they become mixed up. And the good thing is that, when I looked at the actual synteny on the genome, I saw exactly that they do indeed have a very separate parts on the genome where you can even figure out the pseudogenes for each of the respective subclass. And so that means that this separation is true. So right now we're working on the allowing all of you to use this visualization. I mean, the embedding part for you will still be a problem, but at least if you figure out how to embed your proteins, you can use our visualizer, which right now it's almost at the end of its completion, so you can use it as it is already. It's slightly buggy at the point, but it works nicely. And yeah, I mean, if there is any take-home message, is that the protein language models as it is, as they are right now, they already have enough of evolutionary information to have the representation mimic fitness function exploration or like sequence function exploration. I mean, of course, right, we're missing extent forms, but I mean, with that caveat, it kind of works. Yeah, thank you.